So central dogma. Okay.
Okay, madam.
So
It
central dogma of the cell, that
most
is which
important,
was
my dear.
proposed by Francis Crick. So instructions in the DNA which converted into the functional product. So here the central dogma of the cell is that is DNA to RNA by the process of transcription, and from the RNA to the proteins. This process it is called as a translation. So this is a central dogma. So here another, that is RNA to DNA also takes place. So it is called as the reverse transcription. So Hargobind Khurana, so he got Nobel Prize in Physiology and Medicine. So interpretations of the genetic code and its function in the protein synthesis. Okay, so the process, that is conversion of DNA to, here the process of that is DNA to mRNA synthesis. It is called as a transcription, and from the mRNA to the proteins, it is called as a translation. So next one is here. So the properties of the DNA. So generally, DNA increase in the absorption of ultraviolet light and hyperchromic effects, that is low pH. So here the denaturation, that is separation of this DNA, double-stranded DNA will take place at increase in the absorption of UV light and low pH. At low pH, that is 1 to 3, and high pH, that is more than 12. So here the denaturation of this double-stranded DNA will occur. So denaturation means that is separation of the double-stranded DNA into single-stranded DNA. It is called as a denaturation. So in the high temperature also, melting temperature also. Okay, so at the cold temperature, that is annealing. That is again doubling of the this DNA strands will takes place. Renaturation. Okay, so next one is. So for this, they got Crick, Watson Crick, they got, and Wilkins, they got Nobel Prize for the identification of this double-stranded DNA helix. So here another point, that is the nucleosides means sugar, that is pentose sugar plus nitrogenous base. It is called as the nucleoside, and nucleotide means pentose sugar plus nitrogen base and phosphate. So generally nucleic acids, they contain mainly pentose sugar, nitrogenous base and phosphate. So it is called as a nucleotide. Whereas a nucleoside means it contains only pentose sugar and nitrogenous base. It is called as the nucleoside. So another point is, so these are the nitrogen bases, that is purines and pyrimidines, adenine, guanine, cytosine, thymine and uracil. So pyrimidines are cytosine, thymine and uracil. So in the DNA, adenine, thymine, guanine and cytosine, whereas in RNA, adenine, uracil, guanine and cytosine. In place of thymine, uracil is present. So first one, measure. So molecules of heredity, generally nucleic acid, they are called as the molecules of heredity, which allow organism to transfer genetic information from one generation to the next. So nucleic acids first discovered within the nucleus, and presence of the phosphate group and chloroplast and mitochondria. Also general DNA which is present in the chloroplast and mitochondria. So nucleic acids, they are the biological polymers. So it contains polysaccharides and proteins. So first measure isolated the DNA in the nucleus, isolated the DNA from the pus cells and named as a nuclein. So here the primary structure of nucleic acids, that is nucleotides are joined together by the phosphodiester linkage between 5' and 3' carbon atoms of the pentose sugar to form a typical dinucleotide. So information regarding the sequence of the nucleotides in the chain of a nucleic acid, it is called as its primary structure. So how they are arranged? So here adenine followed by cytosine, guanine and thymine. So it is the information, it is passed. So next one is the secondary structure, that is double helix model. It was proposed by Watson and Crick. So here there are different types of DNA, A, B, C DNA also seen. So it contains, so A DNA which contains 11 base pairs per turn, B DNA which contains 10 base pairs per turn, and C DNA 9 base pairs per turn, and Z DNA 12 base pairs per turn. So ABC are the right-handed and Z is the left-handed turn. So here the structure, when we observe the structure, so the diameter is 2 nanometers. So the distance between two nuclear, this nitrogen bases are 0.34 nanometers, and one groove, it contains the 10 bases, and the distance is 3.4 nanometers. Okay. So this is about the DNA. So in next class, we will discuss about the RNA. Okay, sir. Any doubts? Sir,
అంటే ఆల్రెడీ
sir.